chair the motor control session with two very eminent keynote speakers. The background to this is in a way that, as I have gone through my career, I've increasingly realized the importance of motor control in understanding why people move with the mechanics that they do. And when I was attending the committee meeting, Carlo, to my right here, had made a proposal that we start to develop a relationship with a motor control and biomechanics working group. And I undertook as the Congress organizer to help facilitate that. And I'll let him tell you about the two meetings we've had. But in the same process, we wanted to have a session on motor control. So I'm highly delighted that our two speakers are here today, and they're going to give their own views of motor control, past and present. So, over to Carlo.
Thank you, Phil. Good afternoon to all of you, and thank you for this wonderful turnout to what is, as I understand it, the first organized official section on motor control in ISB. This is a new dawn for our society, and I am absolutely thrilled that we have arrived at the point where we recognize that motor control is not just a notion, but it's really a significant part of what constitutes the making of biomechanics. Your attendance here testifies to that, the willingness of the society to sponsor this particular keynote address designated to motor control speaks to that. The support which the society has given us in establishing us as a seed group in 2 years ago, allowing us to hold the first initiative on motor control within biomechanics at the World Conference held in Boston last year, where we had approximately 400 people attending the very first one. Think about it, 400 people showing expressing an interest in motor control within the concept of biomechanics, whereas before there was none organized. 400 the very first time. We were delighted. We still are. So then we continued and had an initiative symposium here on Sunday, and 250 showed up there. Proportionally, even a greater amount than that which showed up at the World Conference, which had about 6,000 people. Here we have less than that. So 1/4 of the members of the attendees, of this conference were in that room. I am gratified. I'm downright ecstatic. I thank you all, and thank you for coming to this first keynote on this topic. It is my great honor and indeed a privilege to lead off this discussion with you. I will intend to take up the first 20 minutes and so, and then my associate, Dr. Farina, will continue. I will give a perspective that will deal mostly with that allowed to an old man, in the sense that I'm going to take some liberties as to how I speak today and extend somewhat emphatically, perhaps, more so than I would otherwise, on some ideas and notions which I have. Please accept them with the warm intent that they are offered. If I question certain aspects of how we deal with issues of motor control, it only is out of affection and love. It is not out of a negative challenge. I want to bring to your attention of some of the people, you people, how I perceive how we now deal with motor control vis-a-vis how we should deal with motor control in the future. And I will try to draw from some lessons from the past and see if putting forward a new notion and schedule will help us to strengthen, make even vibrant perhaps, the notion of motor control within biomechanics. So, with that in mind, I would like to begin. Now, this is about as simple as an expression of interaction between mechanics and motor control that I could conceive. But the point that I'm trying to make here is that when we think of motor control and we think of mechanics, they're really an interacting set that varies to different degrees depending on individual disciplines. So this is an entirely artificial, singularly conceptual notion. It may not even be close to the truth. But I think the relative aspect of these dots are correct, and this is my perception of how the motor control relates to biomechanics. You can see that in different areas, the ratio of interaction between motor control and biomechanics is either greater or larger. But what is important is that when we view this as a whole, this is the space of biomechanics, and it captures various singularized notions of activities, but all within the general space of biomechanics. So that it is important to more respectfully address the knowledge of motor control within the construct of mechanics when we deal with any one of these particular fields. Possibly so to a different degree, but nonetheless to some extent. I think of biomechanics really as a space that covers all of this region here and captures it to different degrees, but essential to all. Now, here comes the bias. You probably all recognize this as a motor neuron, a set of motor neurons beginning in the spinal cord, the anterior horn cell of the spine, traveling down sun nerve construct, reaching a group of muscles, each nerve fiber connecting to a group of muscle fibers, and as a unit forming the motor unit. If I generate an action potential that travels down as it does now, it reaches the muscle, and the muscle twitches as a result. Twitches, it moves. And if I do that repeatedly, the twitches can continue until they tetanize, stabilize in the force output. Why have I shown this? I want to make it clear to all here that the purpose of having motor neurons and action potentials traveling down motor units is so that they can activate muscle fibers. For what purpose? To generate force, not electricity, force, mechanics. The mechanism that determines the production of muscle force should be of fundamental interest to the biomechanist. And as a group of biomechanists, we simply have not addressed these questions at this fundamental level. And yet, this is a fundamental unit of force production in the human body. We need to understand far more clearly and far more deeply than we do so far how this process works, and how it makes us do these wondrous things we do with our muscles so we can express ourselves, interact with our environment. And I remind you again that the purpose of a motor unit is firing is to generate a force twitch, not an action potential. And up to now, these kinds of ideas and thoughts have more or less been the purview of the neurophysiologists. And I think they have done well but I also believe that they have struggled. Their struggle becomes, their mindset comes from a different background than ours does. And I think if we joined them and combined our analytical experiences and our perception of why and how we should study the motor unit behaviors, we'll do together a far better job than has been done so far. Now, I'm going to give 2 or 3 examples as to where I believe that there's obvious place for biomechanists to play, and we haven't, at least not very much, certainly far too little. And I'm just going to leave it to your thoughts to ask yourself questions. Why not? Why don't we understand more about some of these things that we're going to talk about? So think of the following. Most of us, and I certainly among you, when I think of a collection of muscle fibers that forms a muscle, I think of a bunch of parallel fibers, all more or less contracting in the same direction, beginning on one end of a tendon, one muscle, and ending at the other. Well, that's nice and convenient, but we know that that is not the whole truth. But yet, when we talk about modeling forces within the muscle fibers, we generally, for the large part, think thusly. What about cases like this? What about your deltoid, which looks more like the second, which far more this fiber arrangement than that? This hasn't been studied very much. And why not? It's important because the way force sums in here and how heat dissipates and friction is distributed inside here is very, very different than here. So if you're going to be talking about the net force output at the ends here, with this particular fire construct, muscle fiber construct, you have to involve different mathematics and different equations than you do here. What about this case? Does this even exist? Well, in animals, people have published data that says yes. That is, a fiber does not necessarily begin at one part of a muscle and then end at the other part of a muscle. They may, in fact, tie into each other. Think of the complexity that introduces. Think of the amount of work that's been done trying to understand that, close to zero. I mean, can you imagine fibers pulling in opposite directions? How do you control the individual fibers so the forces generally add up? How do you subtract, negate forces that occur? These questions have not been asked. There is one reference which I have at the bottom, which you can see there below, but all it goes back to 1987. Man, I was young in those days. We need much more work done on this area here. What about synergies and antagonists activities? You know, the peripheral and central nervous system have worked out arrangements where over the evolutionary period, they have figured out that if I need to make a movement like this, there's a group of muscles that need to work together. And it's wise if, in fact, over evolutionary time, there's some interaction amongst these muscle, quote, synergistic interaction among these muscles here, so that as they work together, they make the business of producing the right forces at the right time, so I can position this spot and X, Y, Z point in space correctly. How much of that do we know? Neurophysiologists have looked at some of these questions here, but as biomechanists, if you intend to make models to determine what the force distributions of the muscles ought to be so that you go from here to here, why don't we know more about how these synergistic arrangements complement themselves and use them to reduce the decision space of the equation that come out of our models, thus simplifying the solutions and making it more easier to understand how the complex business of generating force from various muscle actually occurs. Here's an example. You may have two muscles working in synergy, muscle 1 and 2. You add and you get the two muscles. Well, that's easy enough. So, but what if you do the same task with synergy M, where you have a bunch of other muscles? Why does that happen? Does it happen? What are the rules? Why don't we know them? One way to, of course, obtain this information is to look at the force packages, if you will, that comes from the different muscles. And that is conveniently done through the use of electromyography by looking at the amplitude of the EMG signal and the on-off time duration of the signal, telling us when the force from the muscle begins and ends, and relatively speaking, how it compares to other forces. There are simple techniques for these investigations. What we need is to excite our curiosity level a little bit. What about cases where you have relatively simple biomechanical models, such as the case would be here? The solution of what the force output, say at this point right here is, depends not only on the fulcrum and the radius and the distance and all that sort of stuff, but also on the intensity and the speed of the movement. How much do we know about the contractile properties of different muscles, and how that influences the speed and the movement, and how that influences the decision space for those equations? Not much. Do we have to? Yes. If I do this, for example, I'm using the biceps in the front, triceps in the back. The fiber type of distribution, the triceps, is dramatically different than that of the biceps. It's much wider than in the biceps. It's not accidental. I don't think we spent, millions of years evolving that way because it was good color coding. So we haven't studied that enough. There are some works, two of them references at the bottom here, one by Walter Herzog and the other one by Bindings et al., who address these points. But this work needs to be enriched. We need to know more. Let me give you some other example. How is a motor unit controlled? That is, to fire. That initial example which I gave you, those pulses. It says, well, we know that there's a recruitment order of progression motor units. You probably all heard of the Henneman size principle, starting out from the smallest motor unit to the bigger and to the bigger and so forth and so on. Well, in 1984, a slightly forgotten gentleman, Seyfarth, produced some interesting result. He looked at the way these fired as they were recruited during an increasing force, and he saw something like this, which we have recently called the onion skin. We have made many observations of this in the latter 2 decades, but I really sort of genuflect to Seyfarth because he did some beautiful work, which I believe is still correct. And it was greatly disregarded for 35 to 40 years in the literature. And what he said was the early motor units, the blue one here, fire faster than the next recruited and the next recruited until the last motor unit fires the slowest. We have called this the onion skin. After Seyfarth, for a period of about 40 years, there was a host of publications that purported to show exactly the opposite. That is, the first motor unit fires slower at any time, and so forth, and the last motor unit fired the fastest. Now, how can it be? I mean, this is simple stuff. We're not talking about really complicated or sexy stuff here, all right? It just doesn't take all that much to get this right. So how come for 75 years there's still a disagreement? You may say, well, who cares? Well, you should care. Because if you calculate the firing rates of these individual motor units, it would be something like this in this case. Here's the blue one that fires faster, that's this one here, and so forth and so on. And if we do so that for the other scheme, It's just the opposite. Okay, says, well, so what? Well, here's what. If you derive a model to calculate the force that is generated from each of these motor units with this firing scheme, as opposed to each of these motor units with this firing scheme, here's what you get. And these are different. And one of these systems says that you generate force very efficiently near the beginning, that is, this force rises quickly with the low threshold motor units, and then when you get to the maximum force level, it becomes a bit unstable, whereas this other one, the after hyperpolarization scheme, as we call it, is more or less linear, and produces greater force output than that one over there. Well, this determines how you interact with your environment. This determines how much force you can generate. It determines how fast you can run. It determines how steady your hand is when you're drawing. And if you're in a caveman drawing something 45,000 years a wall to leave us a message to tell us that he wasn't as stupid as we think he was, having a hand that didn't tremble was important. Okay. So these are fundamental points that we're still arguing in 75 years. And it ain't that difficult. So why haven't we? Well, I'll try to get to that point. So how do we move forward? How do we get past some of these lack of interests of what I thought I pointed out was relevant sort of stuff for biomechanism? How do we get by these small little tit-tat discussions and arguments which we have with respect to how motor units fire? Well, I think we need to train ourselves and our students a little bit differently than we have so far. I think we have to change the didactics. I think we have to give more to our students in terms of understanding about signal processing. They have to learn the difference between signal and noise. They have to know how to manage. They have to those data. They have to know how to separate the wheat from the chaff. And I don't know if we give enough instructions along those lines. My sense is no. Probability theory. This is a mess. Okay? We simply have to do a better job. We have to describe what is meant by independence, correlation, causality, etc. Association between two things does not mean causality. You can walk along the beach every 6 hours and you can see the tide coming in and out. And your head can be heady and you can start thinking that you have the power to move the tides. Well, I got news for you. You're wrong. That's a correlation. That's not a causality. So we have to identify these distinctions for them. We have to put them in environments where they test these concepts. Statistics, we just simply have to be more rigorous about how we apply them. A t-test cannot be used all the time, and they just have to understand this. In biomechanics, I think this certainly does not fall to anyone in this room. We have to approach it from a more engineering perspective. That we do, and we do well. But I think we need to spread that gospel, if you will, to other disciplines. Here's another point that I think we need to pay attention to. We have to go back more now than as we did before, I should say. We don't do sufficient now to apprentice training. I think when a student comes in a laboratory, we have to teach them to think independently. And if we can't teach them to do that, we should force them to think independently. They should ask questions, but learn to respect the status quo. They should take intelligently calculated chances. Yes, take chances. Take them when you're young. Everybody forgives errors in youth. Make a mistake. Tell them not to be afraid of making mistakes, but learn from them. If they don't learn from them, then they're doomed. But I've seen environments where students are overprotected to the point that they're not activated to take chances, make mistakes, and understand the mistakes. Think about how you learn computer programming. You didn't learn computer programming by reading the book. You learned computer programming by programming, making mistakes, and then figuring out what the hell mistakes were and fixing them. And here's another notion that I think we should bring forward. Emancipation from restraining notions. And the word bias tends to scare everybody. There's nothing wrong with bias. It's how we use it that might be wrong. You have a point of view, don't be afraid to show it. Be biased. Remain rational. Be open to scrutiny and challenge. Remember that a hypothesis is a biased proposition. So don't protect your students too much from expressing a bias. Let them control what happens when they express the bias. That's how they will learn. At least, remember, this is a personal perspective. Next point we should deal with is this business of conjecture and proof. Conjecture. An opinion based on presumptive evidence, but insufficient to provide proof. I should have underlined that. Insufficient to provide proof. A fact is surmised to be true, commonly accepted by like-minded and socially bounded individuals. It's more an article of faith than an article of science. Conjecture is good to generate ideas, but they cannot be allowed to live too long without proof. And where does that come from? Empirically based proof. That's what we need to make our point. I'm going to skip this slide here. I want to end with this one example, which is probably familiar to all of you, but I hope it sort of punctuates the central issues that I was trying to outline. Between the year 300 BC and 1543, the Aristotelian geocentric model of the planets moving in a universe dominated philosophy. It was a conjecture. It was internally consistent logic, but had insufficient empirical evidence. And here's the part that gets me. It lasted for 1,800 years. You imagine people got up every day for 1,800 years and believed that. Nobody challenged it. Those that did had difficulties. The Catholic Church didn't like it, and so forth and so on. Then in 1543 comes Nicholas Copernicus, and he says, nah, we don't have the Earth at the center of the solar system, which is what the geocentric model says. What we have is the sun. We have this on the right. This is the Copernicus model, and this is the Aristotelian model. And he says, if I said it like this, there's a lot of things I can explain with what I see there. Well, that ran into a great deal of difficulty with some mathematician, Dye, and so forth and so on. But it wasn't until a few years later, when a man called Galileo scratched some lenses that were about 4 inches in diameters, read some work made by Dutch lens makers, put together a tube that was about 1 meter long, put 2 lenses, one on one end and one on the other end, one converged, one diverged the light, and went out on a cold night in the middle of February at the city of Padua, pointed that telescope we had a magnification of about 40 and scratchy lenses, and points it at Venus. And what he sees is the shadow or the light emitted by the sun forming shadows on Venus. As the Earth moved and Venus moved, and he saw it changing, and he plots this night after night, and he comes to one singular conclusion that he could come to, and he says, the only way that I can explain the forms of these changes of these shadows on Venus is to put the sun in the middle of the planet's circulatory system, and put Venus between the sun and the Earth. empirical evidence. It convinced everybody. And now, this was the triumph of empirically based proof over conjecture. It started a whole new concept of thinking called the post-Galilean thinking. And it's because of that thinking that when we shoot our rockets towards Venus to circulate and understand the planet, we point the rockets towards the sun, not away from the sun. Empirically based proof. We need more of that. We need it in our field. Neurophysiologists need it in their fields. If we want to understand motor control, let us base more of our ideas and notions on empirically based proof. Thank you very much.
Thank you very much, Carlo. My family motto is, we all make mistakes. The secret is only to make them once. So I fully understand some of your comments. Our next speaker is Dario Farina, who is going to give his vision of motor control, past and future.
Yes, thank you, and good afternoon to everybody. It's a real pleasure to be here, for which I had to thank the organizers for the kind invitation. It's also a big honor, actually, to discuss these topics with Professor De Luca, whom I thank for the opportunity to share the stage with him today. I will, in the next 20 minutes, provide a view on motor control and biomechanics that is necessarily not a complete view, but a personalized view. And I will provide examples to support this view. So if we look historically at the way in which we have analyzed movement, on the one hand, we have the neuroscientists that, in a very detailed way, provide us information about the working principles of single neural cells or small group of neural cells. And we have a lot of knowledge on these topics. On the other hand, and this is the other extreme, we have a large community, which is represented here, studying movement from the functional point of view, from the forces that are produced by muscles and exchanged with the skeletal segments. Now, we also have a lot of knowledge on this perspective of studying movements and very accurate models that allow us to estimate forces exchanged with the external environment with high precision. My opinion is that our main limitation to study and to understand still human movement is that we miss a link within these two ways of investigating movement. And the reason why we have difficulties in establishing this link is that we are challenged by the recording of neural activity at a population level rather than at a single cell level, and especially in vivo, in humans during natural movements. And so we are not able to associate a functional meaning to the cellular mechanisms that ultimately determine a movement. So if we look at the way in which we could interface the humans to record neural information, then we have one very special type of neural cells, which has been also introduced by Professor De Luca, which are the motor neurons. The motor neurons converge at the final common path of the neuromuscular system, meaning that they receive the synaptic input from the entire neuromuscular system and transform it into action potential discharged to muscle fibers. And so the motor unit, which is the motor neuron and the fibers innervated, is a very interesting element because, as I said before, is the interface between the neural processing of movement and the function. The motor unit processes the entire neural coding coming from supraspinal levels, brainstem, muscle afferents, and ultimately provide a neural code, which is the last neural code that we generate, after which this is transformed into function. So for this reason, the study of motor units is particularly interesting when we want to link a neural world and a more functional world. So historically, the motor unit activity has been investigated through means of the surface EMG as an ensemble signal, which provides a population activity, but not at the detailed level of single units. And the surface EMG has been a tremendous tool, a very good tool for a number of studies in kinesiology. However, we have to be aware that it also has limitations, which is the reason why advanced techniques have to be developed. And I would like to show to you some of the limitations of the surface EMG signals with a simple representative example coming from the study of reflexes, since the study of reflexes is one of the measured columns in motor control, measured areas in motor control. So reflexes, as you know, are investigated by stimulating the system with a perturbation that can be mechanical or electrical. And what we are trying to do is to identify the wiring of the system by perturbing the system in certain ways and recording the response of the motor neurons through muscle activity, which has been historically done with surface EMG recordings. What you can see schematically represented on the right is an average rectified EMG that is obtained during a stretch reflex, as an example. So there is a baseline activity, and then you can see some peaks and valleys, and these peaks and valleys are interpreted as inhibitory and excitatory pathways to the motor neurons. Now, if we look at this simple example, here we have a simulation in which a number of motor units receive a stimulus, and here you can see the voltage level of the motor neuron, and when it passes a threshold, the motor neuron discharges. Now, in this simulation, we have an inhibitory potential sent to the motor neurons, and thanks to that, you can see that the action potential are delayed with respect to their normal firing. That's a normal reference response to an afferent input of motor neurons. And you can see the average rectified EMG that shows a valley, and this is correctly interpreted as inhibition, as actually an inhibitory pathway. But you can also see that after this inhibitory pathway, the surface EMG seems to indicate an excitation. Actually, one would interpret that as an excitation that has been done in the literature. But there is no excitatory pathway in this simulation. You can see that this peak is generated by the accumulation of the action potentials just because they are delayed due to the excitatory pathway. So this is one of the examples for which we can certainly not associate, we should certainly not associate the surface EMG directly to the neural information. So the surface EMG is a crude estimate of the neural drive to muscles, and just before coming here, I downloaded a very recent viewpoint by Enoch and Duchateau, indicating in a quite extreme way, but effective way, that inappropriate interpretation of surface EMG impedes progress on understanding the control of neuromuscular function. Now, for this reason, historically, many techniques have been developed to assess directly motor neurons. And this has been mainly invasive in the past, starting from the concentric needle of Adrian and Bronk, and then with many other scientists who contributed to different designs. These are invasive systems that are inserted into the muscle, and they are characterized by being quite selective, which is one of the ways in which it is possible then to extract a single source activity. But being quite selective implies that they can sample a rather small fraction of the active motor neurons or motor units. This limitation has been addressed a bit more recently with the surface systems, a bit more advanced than the classic bipolar EMG recordings, and mainly based on multi-channel recordings with the idea of identifying individual motor units, but in a larger number than it was previously possible with invasive recordings. Now, I will show you this system from my research group as an example. In my research group, we believe that the way to go is increasing the number of channels, both for surface system and intramuscular system, in order to detect the detail of motor unit activation, but at the same time, in a large populations to link to the function. And for this reason, over the years, grid of electrodes to be placed over the skin has been developed. This is an example. And also more recently, we've been working on microtechnology for fabricating wire electrodes that allows to record the signal in many detection sites. We can arrive even to 40 detection sites. And these wire electrodes are inserted with a classic EMG needle, and then this part is flexible and remaining to the muscle. So in this technology, we record multi-channel observations of the muscle electrical activity, both inside and outside. And from this multi-channel observation, we try to decode the activation from the motor neurons. And this now can be done in a quite robust way. And what I'm showing you is very new results, actually just accepted by the Journal of Physiology in these days, indicating a decoding of a very large population of motor neurons. This is just an example, but it can be done in many other cases. These are the activity of almost 100 motor neurons, recorded by placing at the same time a large grid of electrodes over the muscle and multiple multi-channel wires inside the muscle. Now you can see that in this case, you have a very large information about the neural population coding that is sent to the muscle. And now, of course, you can think to link this neural information to function. So presently, we are able to decode the activity of a large proportion of the active motor units during voluntary contraction. This is a strong link to muscle function, and I will come back to this in a moment. But what I would like to indicate is that these possibilities are also opening new views to somehow re-examine some of the principles that were established in motor control and neural control of movement in the past. And I would like to provide you an example of this revisitation by talking about motor unit synchronization, which is a phenomenon observed since a long time, to which I would like to give a new perspective. So motor unit short-term synchronization, or synchronization, is the correlated activity between the discharge timings of pairs of motor neurons. It has been observed since a long time, as you can see from the references, and it is classically quantified by computing cross histograms, which provide a correlation between discharge timings of these two motor neurons. Motor unit synchronization is an interesting topic to discuss in this context because it is very tempting and appealing, of course, to link motor unit synchronization to a functional meaning. And indeed, in the past, this has been speculated or discussed in various ways. For example, you can see an example of a simulation here from the group of Roger and Oka, indicating that high synchronization may imply larger variability in force. Now, within the new view of looking at neural activity from a larger perspective, I would like to question this mechanism of synchronization with at least two arguments. The first is related to the way in which it is classically measured. So it is classically measured by computed correlation, as I say, between this output of these two motor neurons. And the idea is that the peak of that cross histogram is related to the relative power of the common input to those two motor neurons with respect to the independent input. But now if you think at the problem and if you have an engineering background, you will realize that in between this measure, which is the way in which we compute correlation, and the actual input, there are nonlinear systems, which are the two motor neurons. The two motor neurons will distort nonlinearly the signal they receive. They even transmit not additively the two inputs. And so it is now well established in simulation and also theoretically that this association does not hold. It is very well possible to have a very strong common input to those motor neurons and record zero correlation. And that's not because they don't receive common input, but it's simply because they are nonlinear systems that distort the input they receive. We are measuring something at the output of nonlinear systems. So this is the first argument that is related to the way of measuring synchronization. But the second argument is more related to the population view that I was discussing at the beginning. And the population view is linked to the function. So this way of looking at synchronization considers pairs of motor neurons. But why do we care about pairs of motor neurons? In reality, we have a very large number of motor neurons generating force. And what happens when we look at a large number of motor neurons, as here schematically represented? Well, let's say that we have a common input, which is indicated by the yellow arrows, and the number of independent inputs that are entering in these motor neurons. They will discharge their firing patterns, and the neural drive to the muscle basically is the ensemble of all that activity. The ensemble is an approximate sum, which means that it's basically an average of that activity. And as you all know, an average enhances the common components and substantially suppresses the independent components. So at the output, when we look at the neural drive to the muscle, actually we get only common input. It's a beautiful system. It's designed in order to decrease the extreme nonlinearity and noisy behavior of these elements by projecting a common input to many of them. And this is a way to transmit this effectively. If we increase the independent input, leaving unchanged the common input, the output will still suppress this independent input in an extremely strong way. So simply, you don't have independent input at the output. So functionally, discussing the ratio between the common input and this independent input is very questionable. So as I say, this filter is tuned on the common input. And this means that there is a poor association between indices based on the proportion of common input with respect to independent input and force. And this view is based on this population kind of analysis rather than individual cell analysis. In addition to this, of course, by having a population view and the possibility to record from populations, we can also connect this with high fidelity with the force exerted. And if we look historically, there have been a lot of attempts to estimate, and this is one actually still of the main open issue, but also applied concept in biomechanics and in kinesiology, of associating force to EMG as a population measure. And more recently, with the work of David Lloyd and others, we also do that in a bit more sophisticated way, in which we have models, sophisticated models of the, for example, fiber orientation and exertion of forces, and from the EMG, with EMG-informed models, we can estimate force. Currently, we can do that in a beautiful way. We can extend those kind of studies to multi-joints with multiple degrees of freedom, and we can, with EMG-driven models from EMG, estimate joint moments, of course, with a calibration on a subject basis. But now, in this talk that look also a bit at the future, what I would like to go to is to make even a step backwards from the neural side and doing this forward modeling and estimation, not from the EMG as an ensemble population measure, but actually from large population of individual motor neurons, decoded in vivo with the techniques that I have just described to you. So is this possible? Well, certainly we have multi-scale models that cross the borders from a very detailed neural description to a reasonable biomechanical approximation. This is a recent paper by Elias and Kohn, in which they study postural sway in full simulation. So there are no measurements here. And they simulated with an inverted pendulum the subject, and then they simulated all the networks that are controlling that posture. And interestingly, with the simulations, they could describe and predict behavior of motor neuron pools in different muscles. For example, they predict that without any particular a priori information put in the model, they predicted a more tonic activity in the soleus muscle and the bars-like activity in the medial gastrocnemius muscle related to the displacement of the center of mass. Interestingly, they have then verified this model indirectly by comparing with experiments and, for example, predicting with the model the bars-like activation of the gastrocnemius and the timing with respect to the center of mass displacement. So very nice work that shows somehow the link between the neural components and the functional and fourth generation component. But of course, what we would like to do is more. What we would like to do is to go from experimental data, from experimental neural data to function on a subject basis. And this implies going from kinematics data of this type and concurrently recorded neural data at the detailed level of neural drive to the muscle, meaning the ensemble of activation of the motor pool to that muscle, decoded online on a subject basis, and on the basis of that, estimating the force of that specific musculotendinous unit for which the neural information has been decoded. And that from this, being able to estimate the force and also estimating stiffness of the tendon and other properties. And in principle, we would like to extend that to all the muscle fascicles we can record from so that this force can be projected on the joints. And so that from the full neural decoding of this task, we can then estimate joint moments, compressive loading, stiffness of the joint. So can this be done? Well, I will present you preliminary data to close this talk that shows that it is possible to do that. And I think we will arrive at the point in which we will be able to do that in highly functional tasks. And I will show you an experiment in the lab, which is constrained still, but they show the concept. It's constrained to 1 degree of freedom. This is the ankle. And recording from the muscles responsible for dorsiflexion and plantar flexion. So this subject has been equipped with the type of recordings I've shown you before. These are multi-channel recordings in a number of positions of the leg. This would be up to hundreds of channels. For each of these locations, we can decode the neural information in the way that I've shown you before. So for each of those locations, we can obtain the neural activity that is driving those muscles. So we can estimate the neural drive, the ensemble of action potential from the motor neuron pool going to each of those fascicles. So this is not surface EMG, this is pure neural activation. These are action potential of a very large population of motor neurons for each fascicle. And then we can estimate force by using forward dynamics. And by estimating force in each of those elements, we can then project it in the joint. And here you can see is a validation in which you can see the prediction of the model starting exclusively from the joint angle information and the neural information, no other information, compared with what is actually measured by a Biodex device. And I don't have time to show you the result, but you can extend the same result in different velocity of fourth generation, different joint angle, in a number of different conditions. You have every time a different neural drive, and then modeling up to the estimation of the force, and then in principle of the joint stiffness and many other mechanical variables. So this is really the link between the neural world, because now at this level, we can do all the reasoning that the neurophysiologists teach us. We can look at the level of synchronization. We can look at the common input they receive. We can look at the onion skin. We can look at any kind of mechanism. We can see how these impact the actual force generated in natural tasks. So I would like to provide you a summary in order to be on time with the time slot I've been given. And as a summary, I have provided you a personal view of the topic motor control and biomechanics. I've started my talk by proposing to you a problem in our understanding of movement. So the gap that there is between a detailed analysis of the neural mechanism of movement generation and a detailed analysis of the function. This can be done as a link. It can be done with surface EMG, as it has been done classically, and which is unquestionable of high value. But we can do better now, and with modern view, with modern techniques, we can actually arrive really at the neural level, really at the neural cell level. And we can do that with a number of cells decoded, so that that population activity can really have a functional meaning. And the demonstration of that is that if we take this decoding now, we are able only with that information to fully explain function, which is of course a proof that now we are at the level in which we can decode a sufficient number of cells for which we can link this level and this level. And I think with this, in the future, we may start a re-examination of the neural and mechanical control of movement under a neuromechanical approach. Thank you very much for the attention.